This section presents the diversity and community structure of zooplankton in Dila River in Palitaw and Pasugong Bukidnon. A total of next A total of 10 zooplankton species were recorded in Dila River, with a total of 89 individuals were collected. These species were grouped into four major taxa. We have the rotifer, 6 species; copepods, 2 species; Cladocera, 1 species; Regularian, 1 species. Among these, Brachionus species and Thermocyclops crassus are commonly distributed in the Philippine freshwater ecosystem. In contrast, Acarcia tonsa and Regularian species are typically marine or estuarine organisms. Their presence in the river is likely incidental rather than representing a established freshwater population. Next. Next. As shown in the figure, Rotifera comprises the 77% of the total individuals, which follows with the Copepoda contributed a 21%. The Cladocera and Regularia were recorded in a very low abundance. The dominance of Rotifera in Dila River is consistent with studies conducted in other riverine ecosystems. Rotifers reproduce rapidly, have short life cycles, and tolerate fluctuating environmental conditions. These characteristics allows them to dominate under unstable lotte conditions. On the other hand, copepods and cladocerans usually occur at a lower abundance in river because their longer developmental cycles and larger body sizes makes them more sensitive to water, current, and environmental disturbances. Radiolaria were recorded at very low abundance, which is expected since they are primarily marine organisms and only occasionally appear in freshwater systems. This abundance pattern reflects a typical riverine zooplankton structure where small and fast reproducing taxa dominates. Next. At the species level, the zooplankton community was overwhelmingly dominated by Brachyanus species, comprising the 70% of the total individuals, which we have the other species such as Acarcia tonsa, 13%, Thermocyclops crassus, 9%, and several species recorded at only 1 to 2%, were much lower proportions. Strong dominance of Brachyanus is commonly reported in disturbed or nutrient-enriched freshwater systems. This genus is highly tolerant to fluctuating and environmental parameters and can rapidly increase in number, allowing it to outcompete other taxa. Next. Clear temporal variation in zooplankton diversity was observed across the 3 sampling months, in which we have in November a higher diversity of 1.35, in October a moderate diversity of 1.04, and in December the lowest diversity of 0.81.
Now for the correlation of the zooplankton, community structure and water quality in the river Palatay Pasugong Bukidnon. The correlation between the zooplankton community structure based on species abundance, evenness, dominance and diversity, and water quality in the river was analyzed using the principal component analysis or the PCA and is presented using the correlation figure which was used to identify the major environmental variables influencing zooplankton community structure in the river in Palatay Pasugong Bukidnon. PCA highlights the dominant regions that controls ecological operations, where in the closer the variables are positioned to each other implies very positive correlations. In this figure, this shows that the relationship between water quality parameters and zooplankton abundance in the river. The results suggest that zooplankton taxa such as Acarcia, Licana, Thermocyclops, and Brachynes are positively associated with higher connectivity and biology conditions. According to the US EPA, high conductivity and BOD indicates elevated levels of ions and organic matter in water, often reflecting mineral enrichments or pollutions caused by anthropogenic inputs such as agricultural runoff and domestic waste. The close association of this taxa to high conductivity and BOD therefore implies its tolerance to stressed water conditions characterized by high oxygen demand and high conductivity. According to Youssef, these taxa are highly tolerant to organic enrichments and fluctuating oxygen conditions due to their adaptive physiological traits and rapid reproductive strategies, allowing them to persist under increased organic loading. For the case of regular for the case of Regularian, it is positively associated with higher TSS levels, which according to UNICEF likely reflects its associates with turbid waters where suspended particles and microorganisms are abundant. In most cases, elevated TSS greatly affects zooplankton distributions by modifying the ability, light penetrations, and food availability, thereby influencing feeding efficiency and habitat suitability. However, considering the Regularians are marine estuarine species, these are possibly that the strong relationship that is as reflected in the figure shown in the screen may not be due to its ecological preference to high TSS conditions, but rather than hereditary conditions that resulted in increased suspension of solids. There is a possibility that the presence of this taxon in the river is associated with water conditions having high suspended matter, often caused by increased rainfall or water runoff, which may have disturbed the riverbed, allowing the suspension for the marine remains, or incidental introductions of this species to the freshwater ecosystems due to water runoff. The distribution of sampling months in the PCA graph as shown also in this figure further supports the possibility with October is typically characterized by heavy rainfall and is shown to be highly influenced by TSS, thus the increased precipitation in this month resulting in disturbance in riverbed and increased water runoff may have positively centered incidental documentation of this non-riverian taxon. Similarly, the presence and abundance of zooplankton in the river is an indication of an ecologically functional and more moderately productive freshwater ecosystems during the October to December sampling period. The dominance of rotifers, particularly Graphines sp, together with the continued presence of the copepods and Cladocerans, shows the river's capacity to support diverse zooplankton groups despite seasonal hydrological variability. This assemblage pattern suggests that quality conditions remain generally acceptable for sustaining aquatic life, as both tolerant and ecologically significant taxa were maintained throughout the study period. Overall, the zooplankton community structure provides strong biological evidence supporting the Tila River's ability to sustain favorable ecological conditions during this study period, and this shows the effectiveness of zooplankton as bioindicators for the river's health.
Next fact. Next fact. Hindi ako dali ay. Yeah. The PCA revealed a clear seasonal patterns in the zooplankton community in relation to environmental factors. In October, high total suspended from, from heavy rainfall increased water turbidity, limiting phytoplankton growth and food availability, which resulted in a more even but less diverse community with 1.04 diversity as no single species could dominate. In December, elevated conductivity and BOD created a stressful condition favoring only tolerant species such as the Acacia tunsa, Cosmena fatalis, Thermocercocrassus, and Brachylos species, leading to high dominance and lower diversity with 0.81 diversity. These changes illustrate how ionic and organic pollution can reduce species richness and alter community structure. In November, however, show optimal conditions with high dissolved oxygen and moderate temperature, supporting multiple zooplanktons and resulting in the highest diversity with 1.35 diversity. Favorable water quality promotes species coexistence, metabolic activity, and reproductive success, indicating a stable and reproductive environment for zooplankton in the River.
For this part of the result, I will be presenting the cluster analysis of the zooplankton community across these three sampling periods, the October, November, and December 2025. As shown in the figure, the cluster dendrogram reveals a clear separation in zooplankton community structure. Among the sampling months, notably, October and November are grouped closely in one cluster, indicating a high degree of similarity in species composition and relative abundance between these 2 months. This clustering pattern suggests that the zooplankton communities observed during October and November were generally comparable. Specifically, both months recorded the presence of Bracchianus species. In October, 2 individuals were documented, while in November, the abundance increased to 11 individuals. Although there was an increase in population size in November, the occurrence of the same dominant species in both months indicate a similar community structure. Furthermore, both months recorded a Shannon diversity index value of greater than 1, which suggests a moderate diversity and relatively stable ecological conditions. Next. The similarity between October and November can be explained by the nearly identical physicochemical condition recorded during this sampling period, as shown in the figure. Water temperature remained almost constant with 24.9 degrees Celsius in October and 25 degrees Celsius in November. Conductivity values were identical at 89.33 US centimeter, while biochemical oxygen demand or BOD remained low, measuring 1 milliliter, ano na siya? Milliliter per liter and
Kaya siya really,
mg per liter in October and
really
mg per liter in November. The consistency of this these parameters indicates that the river experienced minimal environmental fluctuation during these months. Such stable conditions likely supported the continuous presence and stable distribution of zooplankton species. In particular, the low BOD values, which fall under the good to excellent classification based on the Water Quality Index standards, indicates low organic pollution and sufficient dissolved oxygen availability. Meanwhile, the stable conductivity suggests a relative constant ionic concentration and nutrient condition in the river. Several studies support the strong relationship between the physicochemical parameters and the zooplankton community structure. Villarroel and Camacho 2023 emphasized that temperature, conductivity, BOD significantly influence zooplankton distribution and diversity and freshwater ecosystems. Conductivity reflects dissolved ion concentration, which affects habitat suitability for different zooplankton groups. Water temperature influence metabolic activity reproduction and seasonal succession, while BOD reflects organic pollution levels that may favor tolerant species when elevated. Therefore, the clustering of October and November can be attributed to the stable and favorable physicochemical condition recorded during these months. These findings demonstrates that minimal environmental variation promotes the presence of similar zooplankton community structure in the river.
So in summary, this study evaluated the water quality and zooplankton community in the river Limpaltupas from Bukidnon by analyzing the selected physical-chemical parameters identifying the zooplankton species present in the river. Temperature, total suspended solids, dissolved oxygen, biochemical oxygen demand, and conductivity were measured at upstream, midstream, and downstream stations Over the three sampling periods from October to December, the water quality index was used to assess the overall water quality, while the dissolved plankton compositions, abundance, and diversity were examined to determine their relationship with the physical chemical conditions. And a quantitative research design was applied, and both water and soil samples were collected and analyzed using standard laboratory procedures and taxonomic identification methods. The result showed that most physicochemical parameters were within acceptable standards for freshwater ecosystems, and the salt oxygen levels in October were below the recommended minimum indicating for optimal conditions, but values increased markedly in November and December, reflecting improved oxygenation. Conductivity and pHs remained within the normal permissible limits, while BLD values indicated low to moderate organic pollutions and the capacity for natural self-purifications. Water temperature was also within suitable range for tropical freshwater systems, suggesting stable environmental conditions without thermal pollution. And the computed WKF values indicated that the river water quality ranged from good to excellent during sampling period. And also the zooplankton analysis identified 10 species from 10 species from major groups such as the rotifers, caducerans, copperpods, radiolarians, with rotifers particularly brown pinus being the most abundant. Diversity analysis showed high species diversity in November, suggesting favorable environmental conditions that supported the relatively even distribution of species, and differences in species composition and abundance among stations and sampling months reflected the influence of physicochemical factors and seasonal variations. And overall, the findings indicate that the Gila River maintains generally good water quality and supports a diverse zooplankton community. The study highlights the important role of physical chemical conditions in shaping plankton distribution and provides baseline information useful for future monitoring, conservation, and management of freshwater ecosystems in the riverine area.
In conclusion, the water quality of Dila River in Palotay, Pasig, Bukidnon was generally within acceptable freshwater standards, with most parameters stable and the soil oxygen improving after October, resulting in good to excellent WQI rating. 10 zooplankton species were identified, including rotifers, cladocerans, copepod, and the radiolarian, with brachyla species being the most abundant. Third is that zooplankton diversity was highest in November, indicating balanced species distribution and favorable environmental conditions for their survival. Last, water quality strongly influenced zooplankton composition, abundance, and diversity, with variation across station and mass, reflecting environmental and seasonal changes.
So, for the recommendation, based on the findings and conclusion of this study, I would like to highlight three key recommendations. First, the data from this research can serve as a baseline information for the Department of Environment and Natural Resources, local government units, and community stakeholders. It can guide environmental monitoring, policy development, and conservation efforts to maintain the health and ecological balance of the river. Second, future studies should focus on how rainfall patterns, water flow, and nutrient inputs affect zooplankton communities, particularly during periods of low diversity and abundance. Understanding these environmental drivers will help explain changes in species composition and distribution. And finally, long-term monitoring and comprehensive sampling of both zooplankton and water quality are recommended. Sustained observation will provide a stronger baseline data to support effective conservation, biodiversity protection, and sustainable management of the river. This recommendation aims to strengthen conservation efforts, improve ecological understanding, and ensure the sustainable management of the river. And that is all. Thank you.